Hi everyone. Yes, I'm going to be talking today about my research into population genetics. So I should start by thanking my PhD supervisor, Rachel Fuster, who contributed a great deal to this work, also our collaborator, Emma Carroll, and also thanks to Paul Morrell for his help with the R grid package. So I'm going to be talking today about one of the main questions in population genetics, which is how much genetic connectivity is there between two populations of animals in different places. So as a motivating example for this, the Department of Conservation in New Zealand has spent many years eradicating rats and other pests from a lot of the offshore islands around New Zealand. I'm going to talk today about three little islands called the Broken Islands, which you can see highlighted in the orange box. Those are off the coast of Aotea, also known as Great Barrier Island, which in turn is off the coast of North Island, New Zealand. So when DOC are planning an eradication attempt, it would be useful to know in advance whether the rats are happily swimming back and forth between the island and the mainland, because rats unfortunately can swim quite well, because in that case there's not much point attempting eradication if they're going back and forth regularly, or whether only one or two made it across the island and have populated the island from there. And we can't observe this directly, but what we can do is we can look at genetic samples to see whether there's any kind of link between the populations. If you have populations that are separated for a reasonable period of time, they become more genetically distinct, whereas if they seem to still be genetically similar, then that suggests there's still a lot of ongoing movement between those populations. So where do we start? Well, we're going to need some samples from each of the populations, from the mainland population and the offshore island, before the eradication. And then we extract DNA from these and look at many different points on the genome known as loci. And at each locus, there will be different allele types that you see in the populations. And in this case, we've got a locus with 3 different allele types. And you can see that they've got quite different frequencies in those two populations. So once we've estimated the allele frequencies in the populations based on those samples, we can then look at an individual genotype and calculate what is the probability of getting that genotype based on the estimated allele frequencies in that population. And we can do this for all the different loci and then sum up all the log genotype probabilities to get an overall log genotype probability for each individual and each population. And this indicates, this is a likelihood, so a log likelihood, so it indicates the individual's fit to the population. So this is all well-established methodology implemented in a software called GeneClass. And this is the output of GeneClass. You get a lovely table full of lots and lots of data, but it's kind of difficult to see what the overall picture is from this. So the first chunk of my PhD was working out how we can produce some pretty plots. It's not quite as straightforward as it might seem, but the basic idea is that the log genotype probabilities for the broken islands are on the x-axis, and the log genotype probabilities for the Aotea population are on the y-axis. And each of these little points is an individual rat. So the first thing we can see by looking at this plot is we can see that these two populations, rats found in Broken Islands and rats found in Aotea, are reasonably well separated, apart from this stray little rat over here in blue, which was found on Aotea but looks like it was actually born in the Broken Islands because it's clustering in the Broken Islands population. So apart from that, they're pretty well separated populations. The other thing I can tell from this is that the Aotea population fits well to Aotea in that they are high on the y-axis, but they have a reasonably poor fit to the Broken Islands, in that they're low on the x-axis. The Broken Islands rats have a high fit to the Broken Islands, as you'd expect. They're high on the x-axis, but actually quite a lot of them also have a good fit to Aotea, and that they're also high on the y-axis. So we've got an asymmetric situation between the two populations. And the reason this occurs in this particular case is because almost certainly the population of Broken Islands was founded from Aotea. So any alleles that we find in the Broken Islands were originally from the Aotea population. And so any rats that from Broken Islands that go back to Aotea would fit right in. But the population of Broken Islands has lost a lot of the genetic diversity of Aotea. So a lot of the rats from Aotea will have alleles that are no longer found on the Broken Islands. So that was the first stage of my PhD. And the next question we asked was, okay, we've got this plot with all the individual rats. Can we get some kind of summary of the overall picture for these populations? Could we, for example, think about all the possible genotypes that could arise from a population and then assess the sort of distribution of all their fits to the population? And we can, except that this distribution that we're trying to characterize turns out to be quite awkward. For starters, it's the sum of the distributions at all the loci, which makes it difficult to express analytically. The worst bit is that it's really, really skewed. When I say it's really, really skewed, I mean that on the log scale, it's still incredibly skewed. It can cover hundreds of orders of magnitude. So it's the most skewed distribution you will probably ever encounter. And the way we got around it was to end up approximating it using the saddle point distribution method, which is a really handy approximation method. And I won't go into the details now, but effectively it lets us augment these gene plots with some additional auxiliary plots. So let's look at the bottom plot now. So on this bottom plot, this orange line here is showing the distribution of all possible rats that could arise in the Broken Islands and how well they would fit to the population of Broken Islands. The blue line at the bottom is the fit of all possible rats from Artia and how well they would fit to the Broken Islands. The key thing to note is there's very little overlap between these two curves. So this indicates that almost none of the rats from Artia would fit well into the Broken Islands. If we look at the left-hand plot, this blue curve is the fit of all possible Artia rats into Artia. And then the yellow curve is the fit of all possible Broken Islands rats into Artia. Now we have a lot more overlap on this side because actually quite a lot of the Broken Islands rats would fit well into Artia. So you can think of these sort of auxiliary plots sort of like cross sections through the central gene plot, parallel to the X and Y axes. But you can see clearly from these side plots the fact that we have this distinct asymmetry between the populations. Let's look for contrast at another pair of islands, another pair of locations. So Kaikoura is another little island off Artia. Now here we can immediately see from the central gene plot that these two populations are a lot more overlapping than we had for the Broken Islands and Artia. And Artia has much more overlap into Kaikoura than Artia had into the Broken Islands. And Kaikoura has a lot more overlap into Artia than the Broken Islands had into Artia. So all in all, there's a lot more overlap going on. And this, as it turns out, is because the Broken Islands have little cliffs to stop the rats turning up, whereas Kaikoura has got a nice little beach for them to land on, which is really not helpful because they happily swim back and forth and back and forth and back and forth. And effectively, there's no point in trying to eradicate them from there until you can eradicate them from the whole of Artia. However, even though you've got more overlap in general, you can still see what might not be obvious just from the central gene plot alone, which is that again you've got this asymmetry, which is again due to the fact that Kaikoura was founded from Artia. So another benefit of having these sort of summary plots is that you can also then get numeric measures to summarize the situation between populations. We've actually got 3 of these measures. So the first of these measures is simply the area of overlap between the curves in the side plots. So we've got an area of 0.02 overlap in the bottom plot, very little overlap with baseline Broken Islands, and much more overlap with baseline Artia. The Broken Islands rats would still fit pretty well into Artia. This is reasonably intuitive to explain visually. It makes sense, well, we found to biologists to show it visually, but it's hard to explain what it means. So we've also come up with another couple of measures, which I'll explain the first one, called incumbent selection probability. I'll explain by means of an analogy. So imagine that you have two children in front of you. One is Dutch, one is English, but you don't know which is which, so you give them an English language test to find out. The incumbent selection probability is the probability that the English child will be better at the English language test than the Dutch child, and therefore the English kid will be correctly selected as the incumbent in that population. Now, it should be noted that quite a few Dutch kids speak pretty good English, so there's actually a reasonable chance that they might actually do better at the English language test than the English kid. If you go the other way, very few English kids speak Dutch, and so it's highly unlikely that an English kid is going to be better at the Dutch language test. So we look at this our example of broken irons, it's almost certain that the broken irons rat would have a better fit to the broken irons than the Artia rat would, but it's not always not certain that the rat from Artia would do better in Artia. The final measure is called the home assignment probability. So the other one was looking at two individuals and one population. This is looking at one individual and two populations. If you've got one individual and you don't know where to assign them to, then you can give the child the English test and the Dutch test to find out which population they came from. And the home assignment probability is the probability they will do better at the English language test and therefore be correctly assigned to the English population. And so here we've actually got a probability of 1 in both directions. So actually the broken irons rat is almost always going to have a better fit for broken irons, and the Artia rat is almost always going to have a better fit to Artia. Finally, because the numeric measures, because of the numeric summaries, we can also then do permutation tests in order to actually test the significance of these measures. So I'll just quickly finish with one little extra example. So this is from our collaborator Emma Carroll, who's done a lot of work with different cetaceans, particularly southern right whales. So southern right whales occupy various places around the southern oceans, and I'm going to focus on the population of the Indo-Pacific populations around New Zealand and Australia, and the southern Atlantic populations near South Africa and Brazil and Argentina. And there's also this little population here around the South Georgia Islands. So you can plot all of the different populations in a gene plot. It's actually feasible to plot more than 2 populations in a gene plot. Currently what we do is to take PCA of the log genotype probabilities and then plot the first 2 principal components. But we might well also adapt in future to use Dan Cook's wonderful little Tor software package. And we can then also look at individual populations. And again, we can see we've got asymmetry between Argentina and South Africa, where the Argentinian population is almost entirely overlapping the South African population, but the same is not true for the baseline of Argentina. And finally, Emma was particularly interested in identifying the source of additional individuals that she found around the South Georgia Islands and near Chile. And so this is showing the population of the Indo-Pacific superpopulation and the South Atlantic superpopulation. And the individuals in orange, the extra individuals, are the ones from South Georgia. Sorry, and we can see that they are probably from the South Atlantic superpopulation. And finally, I should say that we've got an R package for this up on GitHub, and there's also a Shiny app. And thank you very much.